We'll move to the next talk, which is given by Professor Annapurna Vyakarnam. She's a group leader, HIV/TB co-infection at CIDR, Indian Institute of Science. In addition, she's also a reader in microbial immunology at King's College London. Dr. Annapurna, please get going with your talk. You might want to share, Annapurna. Hello, Annapurna, are you there? Annapurna, are you there?
She has to unmute herself.
Just hold on, I'll try to call her.
Okay, fine. I don't know whether Annapurna is having some problems in logging in. We'll just need to be patient for a few more minutes.
It will take about 5 minutes. We'll be able to fix this.
Okay, all right. So if there are any other points you want to discuss, Shashank, this might be a good time while we...
We just need to stay on time because following up, we don't have any lunch break. So we need to finish on time. So we have to stay on time.
Fine.
Only break we have is about 15 minutes tea or coffee break. That's the buffer time. If anything spills over, we can cover it.
Right.
That's 45 to 5 p.m. Yeah.
Right.
So can I ask a question? Yeah.
Yes,
Sure.
Why are registrations closed? It's an online meeting. No, anybody should be able to attend.
No. Okay, we see you now. We see you now. Let's turn off the phone. Okay.
Can you hear me?
Yes, we can hear you. We can see you. Your slides are up.
Perfect. Okay. I'm so sorry about this slightly delayed start. Shall I go ahead?
Yes, please.
Yeah, go ahead, Annapurna. Go ahead. Yeah.
All right. Okay, thank you very much for this invitation. I think it's so appropriate, and I've really enjoyed the session today. And what I'm going to try and perhaps place in context is this dramatic increase and interest in trying to use BCG vaccination in the context of COVID-19. So if one just looks at the number of clinical trials that are currently in play in the context of COVID-19, the vast majority of them are targeting are specific vaccines meant to induce a specific immune response to the virus itself. So based on the concept of inducing memory responses to the virus itself. But there are a group of vaccines now, of which BCG is one, where one is trying to boost immunity to COVID-19 through the concept of boosting heterologous immune responses. What has to place, I just want to highlight this news which came out yesterday from Pfizer and its company, other company, BioNTech, about the potential to use the potential of vaccination in the context of COVID-19. This has made big news, and I think there is one very heartening point that we can take home from this. And this is the first evidence that that if one were to induce an immune response against COVID-19, it could lead to protection. All the previous vaccines, all the vaccines that are in trial, including the Oxford vaccine, which is fairly well advanced, does not have that data. What several vaccines do have is they have demonstrated that the vaccine itself is capable of inducing an immune response, that is, i.e., it is immunogenic. But the concept that that induced immune response can confer protection and can reduce incidence rates or disease has not yet been demonstrated, simply because in the context of the Oxford vaccine, which is very well advanced, we simply do not have the numbers of infected individuals in the vaccine arm to make the call. But this RNA vaccine, where one is giving nascent RNA, getting the RNA to be translated in vivo, express protein, and then for that protein to be recognized by virgin B cells, and then to produce, to expand, to produce to secrete antibodies, and then to have that antibody response and the induced T-cell response to be able to then control infection is something which is extremely heartening, because it does show that we are in a position to induce protective immunity against COVID-19. So this is great news, which has just emerged yesterday in terms of primary evidence for protective immunity. So why BCG? So where does BCG come in? So there are, to the best of our knowledge, as of yesterday, there are about 20 COVID clinical trials of BCG in the context of COVID-19, and they are looking at a number of different clinical endpoints. So one clinical endpoint is to vaccinate healthy individuals and see whether, particularly healthcare workers who are in the front line of COVID-19, and see whether one can reduce absenteeism. Another type of trial is to give BCG to high-risk elderly individuals, albeit the vast majority of these trials are in TB non-endemic regions where BCG is not given at birth, quite unlike India. And in these individuals to see whether it potentially reduces morbidity and mortality. Another type of trial is to give it to just healthy volunteers and see how many of them will become infected. So there is a combination of trials which are in play at the moment, with the large randomized clinical trials looking at primary clinical endpoints in terms of either hospital admissions, disease severity, or in terms of protection of incidence of disease. So is there any evidence that it can, in fact, work in the context of COVID-19? Can BCG actually be effective in the context of COVID-19? The short answer is we do not have enough data to make that call. The long answer is there is some data which has been published recently from a retrospective cohort study. So this is a group of individuals, again, in a TB non-endemic region of the world, who were given BCG well before the COVID-19 pandemic took over. And what they're looking at is how many of these individuals in the context of COVID-19 at a significant time before the pandemic, and asking how many of these individuals either became infected or to look at disease severity. So the N here is probably not significant enough to make a call on a disease incidence, that is incidence of infection. But what they were able to report is a small reduction in one of the clinical symptoms of COVID-19 infection, which is fatigue. So some early data, and which has come out in my own mind, this is premature evidence. So, but nevertheless, there is this data which is out there, which talks about the potential for BCG in COVID-19 as being the only study so far which is published. There is other epidemiological data which has been extrapolated to infer that BCG may well have a protective role, with perhaps the most telling data coming from two countries which sit side by side, in this case Portugal and Spain, with Portugal, which has a mandatory BCG vaccination having lower population incidence of COVID-19 in lighter pink compared to Spain, which is at a much higher level of incidence of COVID-19. And metadata through the world would indicate that if there is a mandated policy for BCG vaccination, those countries in the world are reporting certainly lower deaths and maybe even lower cases. But this is only epidemiological extrapolated data. So what is BCG and how is this potentially of interest in the context of COVID-19? Well, so BCG is basically a vaccine which is the only vaccine which is being used in the context in the fight against TB. And it's basically a Mycobacterium bovis, which has been, it's a live attenuated vaccine, which has been passaged several times over and has accumulated major deletions in the genome, with the most profound ones being in the RD1 region. And therefore, BCG, the vaccine that is given, lacks certain virulence factors like ESAT6 and CFP10. And these and continuous passage has led to these significant deletions associated with attenuation. And it's given and seen, and its efficacy is most profound in children, where it protects against mealy TB and TB meningitis. And I'll talk a little bit about the potential of BCG in the context of adults. But it has been found to reduce child mortality by offering protection against several life-threatening pathogens, and these are unrelated pathogens. And this, I think, underpins by far one of our biggest interests in the context of BCG in the context of COVID-19. So how is it working? BCG, we believe, can work at two different levels. So first of all, it is a pathogen which is, or a microbe, which is readily taken up by the monocyte macrophage lineage of cells. It can go into a number of different cells, but particularly in the macrophages, it finds a home there and is able to activate the macrophages significantly by engaging a number of pattern recognition receptors, which includes TLR2, TLR4, the NOD2. It can engage with DC sign on macrophages and DCs. And this initial activation is absolutely critical to how BCG, we believe, through subsequent data, which I will just briefly touch on, is able to impact overall immunity. We believe that it can also, by enhancing antigen presentation, also work to activate the adaptive immune response. So BCG can work both at the level of innate immune cell rewiring and activation, as well as adaptive immunity. And therefore, your initial phase, which happens of innate immune activation, is followed by activation of adaptive immune cells. So it is this concept which is being used in the fight against TB itself. And one shouldn't forget that BCG now is being seriously, is now seriously in the running as a vaccine for TB itself. And this is based on emerging data. So for several years, BCG has been around for 100 years, and we know that it doesn't confer lifelong protection. We also know that it doesn't against TB, and we know that it doesn't confer prevent the augmentation of TB as individuals progress to young adulthood. It gives a certain level of protection in the context of the young, but it doesn't give protection in the adult. So the question has emerged in the absence of other efficacious vaccines is, can one probe BCG in the context of TB to revaccinate individuals as young adults and induce adaptive responses that may be pertinent to control of TB disease. So we've contributed to this area. This is data which has been published, and this is a first of its kind study where we were able to do in-depth immunology on individuals here in India, where the question is whether it is safe to give BCG to those who have already been vaccinated at birth. And we addressed this comprehensively. We took individuals who are either latently infected with MTB or not latently infected, gave them BCG in the ages of between 18 to 24, exactly that age group which is most vulnerable to TB with time. And we were able to demonstrate that this revaccination can induce MTB specific, that is antigen specific T cells in these individuals, and it is safe with no adverse reaction following revaccination. We were also able to demonstrate that we can alter the balance of the CD4 T cell response in terms of key cytokines which are expressed by these cells, IL-17 and IL-10. And we were able to demonstrate that revaccination in these young adults is able to induce a population of CD4 antigen-specific T cells, which we call regulatory T cells, which are important in dampening inflammation, so-called regulatory IL-17 positive cells in vaccinees compared to non-vaccinees. And our data came out at about just the time when BCG was gaining interest in the context of TB protection. And this very elegant study from the macaques showed that if you are able to give BCG and then do a live challenge in these macaque models, you get reduced incidence of disease and CFU in the primary lobe, in dissemination, in the bowel, and in the lung, the draining lymph nodes. There is reduced CFU in the BCG vaccinated animals, particularly those that have received BCG through the mucosal route. And this is allied with the induction of key cytokines. So what the data shows, interestingly, that if you give BCG or you give a control, you can induce interferon gamma, TNF alpha, and IL-2. But what distinguishes these different groups is this capacity of the vaccine in the mucosal route to induce IL-17 with the best level of protection. And that is also, in addition to 17, one is also inducing IL-10, antigen-specific IL-10, which concurs with our study of the potential importance of this. And finally, this is perhaps the most telling data from Bob Ceder's group, where they were able to give BCG intravenously, which in itself is controversial, but this is elegant data, which shows that if you give BCG intravenously, you almost get 100% protection compared to the other routes. So BCG is now re-emerged as a key candidate in the control of TB. So just to put in context, BCG is being looked at, in the context of autoimmunity, where it is being used potentially to dampen inflammation, in the context of cancer, where it's being used to enhance immunity. It's in respiratory infections, where it gives heterologous protection, and neonatal sepsis. So how is BCG potentially mediating these heterologous protective effects in several diseases? The number of the mechanisms really do vary quite a bit. So in the context of neonatal sepsis, it promotes granular poiesis, so it increases neutrophil numbers, which is extremely important in that first line control of microbial killing. In the context of bladder cancer, it is able to activate tumor-infiltrating T cells, thereby improving anti-tumor immunity. In the context of EAE, it is able to alter the balance of Th17 Treg frequency. So clearly, the regulation of Th17 is context dependent. In the context of type 1 diabetes, it is able to induce the secretion of IL-10 by B cells, which is important, which is clearly important. So how is BCG mediating these effects? This has led to the concept of trained immunity. So we all appreciate that memory is imprinted in the context and is a feature of the adaptive response. So this concept that innate immunity or innate cells can also have an imprint of memory was first introduced by Nettier et al., where what the concept follows, or where the evidence is that if you expose certain innate cells to activation via PAMPs, pattern-associated molecular patterns, by activation through those engagement with TLRs, then when the same cells or at the population level meet either the same or heterologous PAMPs, you get a better and more potent response. So that then places perhaps the context of the interest in BCG in regulating perhaps heterologous protection, where in the context of a disease like COVID-19, where we are waiting for a specific vaccine, we could potentially be protecting the population by giving BCG, which could then provide a certain level of non-specific heterologous protection whilst waiting for specific vaccines. BCG also has adjuvant properties, which would also be beneficial for in the context of any vaccine. So what is the evidence that there is this heterologous protection linked to this concept of trained immunity? The best evidence has come from, again, the Nettier group, and I have to say that there is really to date, there aren't a significant number of publications in this area, but it is a very significant and growing field. So the way the experiments are done over here is to expose or to vaccinate individuals with BCG first, and then follow up with another vaccine or another viral infection. In this case, they followed up with yellow fever vaccine, which is a live vaccine. And what they're able to demonstrate is that if you take cells from the vaccinated individuals compared to the placebo, these cells will respond to a number of PAMPs much better than the control. So it will respond to LPS, MTB itself, Candida albicans, and it will do so by secreting key innate cytokines IL-1 beta, TNF, and IL-6. And this is correlated with reduced viremia in these individuals. And this is a key, and this, I believe, is a significant observation that you can get reduced viremia concomitant with the induction of the circulating and innate cytokines at the in this context at the monocyte macrophage level. So how is BCG working to induce trained immunity? So the data at the moment would point very strongly to epigenetic remodeling. So what BCG is able to do is able to alter the level of methylation and to relax the chromosomes so that what we are getting is markers of active gene transcription and active gene expression and augmented gene expression at the level of the macrophage. The other data which would support this is that it not only happens at the level of the mature cells which have emerged, which are circulating, but that this could potentially take place in the bone marrow on myeloid precursors. So the imprinting is followed through and can be persistent or maintained to a certain extent over time. And what one is able to associate this epigenetic a change to is increased protein at the transcriptional level to increased microbial killing, cytokine expression, and adaptive immunity. And this is the last
Annapurna, can you round it up in about 2 minutes or so?
Yeah. Okay.
Thank you.
So I will just go through to, and this is just demonstrating that IL-1 beta is key in this. So in the context of COVID, do we have any data to indicate that BCG could be significant? And I'll show you just some preliminary data from our ongoing BCG study. So we have a BCG study where we have recruited individuals, healthy volunteers, where we have vaccinated them or controls. It was interrupted by COVID-19, but I'm going to show you some preliminary data to show that the monocytes could well be impacted. So first of all, we show over here that IL-1 beta, a key cytokine, is induced. And this is just a small number. This is just about 8 weeks worth of data from this study where we have archived samples now that we are back, which shows a trend. It doesn't reach significance in terms of IL-1 beta is induced. And lastly, that we can look at the macrophages over here and demonstrate that the macrophages that are isolated using flow cytometry are responding much better by secreting a key innate TI cytokine, which is TNF-alpha. So I would like to place this data in broader context. We have, from our retrospective analysis from our previous study, we can demonstrate that the frequency of CD14 positive TNF-alpha is a very dynamic cell population, but we see significant increase in both IGRA positive and IGRA negative vaccinated individuals compared to unvaccinated controls. So we are able to reproduce this. And finally, I won't include the T cell response, but to end with this data, that one of the concerns with BCG is whether it has non-specific broad pro-inflammatory effects. And from a large set of negative data of plasma cytokines, we can say that BCG does not have global activation potential. So with the help of this award, which we've just received over here, which is hopefully will be released soon, this is what we will do. We will now poise to look at both testing the concept that BCG vaccinated subjects will be able to respond more efficiently to SARS-CoV-2 lysate in a direct test. We will be able to look at the epigenetic changes induced in these cell populations and to link that with the transcriptome, both at the bulk level and the single cell level. So I really am very grateful and very fortunate to have a very talented team of scientists with me. Asma Ahmed, who has led some of this work, Shrabanti and Vashishta have been with me for some time and have contributed fantastically. And there have been other MSc students. This is a collaboration between NIH and us, and also between Leiden, where they're working on where we're part of a larger BCG vaccine trial, and St. John's Research Institute. So thank you very much, and apologies for the start.
Yeah. All
Happy
right.
to take
Thank you so much, Annapurna. There are lots of questions, so I'm going to quickly go over them, and if you could answer them very quickly. The first one is, you mentioned that BCG upregulates Treg cells, and these are immunosuppressive. Is there a way BCG can abrogate cytokine storm in COVID-19 patients?
Yeah, so I think it's a great question, and I think that we will definitely be able to track that in a vaccine study. BCG ability to activate Tregs is at not at the level of the progenitor. It is at the level of rewiring by altering the balance of pro-inflammatory and anti-inflammatory cytokines. So whether it is in itself going to be enough to induce significant Tregs, I don't know, but it will certainly be able to induce the level of IL-10 secretion, which has been demonstrated at the cellular level, not at the plasma level, but at the cellular level. So yes, it could have potential impact in the adaptive response. Yeah.
Okay. The next question is, do we know the contributions of CD8 memory cells in trained immunity induced by BCG? So the idea is that strong CD8 memory response induced by SARS-CoV-2 might be useful.
So absolutely. So the question here, underpinning question is, will trained immunity boost? What type of adaptive responses will it boost? So there is no reason to believe that trained immunity will not boost both CD4, CD8, and B cell responses. There is every evidence in the yellow fever virus vaccine that it boosts CD4 T cell responses in that system, but there is no reason but there's no direct evidence that it will activate all classes of adaptive cells, but there's no reason to believe otherwise, so it can be tested. Absolutely.
Right. Finally, does serum from BCG vaccinated people react to SARS-CoV-2 viral proteins?
Not been tested, and I'm struggling to think why that should be the case over here. Because I'm just trying to think why serum from BCG should react to SARS-CoV-2, which is, I think the question maybe maybe I can reframe it. There is fantastic data which has emerged about cross-reactive immunity. And if the question is being directed or thinking, whoever has asked this question is thinking about cross-reactive heterologous protection, that is those who have BCG have elevated levels of their innate and adaptive response is at a higher level of preparation because the cells have been rewired and can respond better to PAMPs, and SARS can of course activate through PAMPs. We know that it can activate the type 1 interferon pathway. Then is there any evidence that there may be some cross-reactive antibody responses in BCG vac? And the answer is people are looking. We can certainly look to circulating coronaviruses, antibodies to circulating coronaviruses, and we can definitely ask whether vaccinated individuals have a better antibody response to SARS-CoV-2. But so that
one more, Annapurna. Can trained immunity be elicited by viral vaccines?
So I think that the question is, how does it work? I think that that a number of PAMPs can activate trained immunity. BCG is not the only one. And in fact, the data from Nitiya's group would show that IL-1 beta per se can induce epigenetic modification in the right histone modification methyltransferases in order to get better responses. Can going through the double-stranded RNA, going through TLR7 induce the same? There is no direct evidence to date, but LPS, BCG, Candida can all induce trained immunity very efficiently. We know that type 1 interferons can certainly promote trained immunity, so I would suspect that it can, but there is no direct evidence.
Great. Thanks a lot, Annapurna. Your talk elicited a lot of responses. Thank you.